Hi everyone, welcome. Thanks for joining us for today's webinar that's organized by the Gene Convening Global Collaborative and Target Malaria. Really delighted to have you participating today to hear a little bit more about some of the work that's gone on to think about the potential first field trials of gene drive approaches to help control malaria. So. Gene drive is a genetic biocontrol approach where through genetic modification of a disease vector like a mosquito, it may be possible to introduce a genetic change and spread it through the wild population of mosquitoes that ultimately reduces the number of mosquitoes or interrupts their ability to transmit the malaria pathogen, and through either of those approaches helps to control malaria. These kinds of technologies have been studied in laboratory environments and contained laboratory studies in cages where there's been exciting results scientifically in terms of the ability of these gene drive approaches to perform as hoped in those settings. But for evaluating these technologies, it's a really important question how those approaches perform actually in field environments similar to where they would be used for malaria control. So as of yet, there has not been any field trial or any release of a gene drive modified mosquito. There have been field trials of genetically modified mosquitoes that don't carry gene drives. But looking ahead to potential field trials for gene drive approaches, there are some additional considerations beyond those associated with mosquitoes that are, for example, genetically modified to be sterile. The Gene Convening Global Collaborative supports stakeholders to identify and address key questions associated with the development of these genetic biocontrol approaches, including gene drive for public health, particularly for malaria. And we are happy to have had the opportunity to convene a working group of a number of the researchers involved in considering how to potentially design initial field trials the initial field trials of gene drive modified mosquitoes for malaria. And so that group, which included participants from Target Malaria, from the University of California Malaria Initiative, from Transmission Zero, and pulled in many different experts from many different domains, had the opportunity to come together and develop a journal article on considerations for the first field trials of low threshold gene drive for malaria control. We wanted to take the opportunity in today's webinar to share some of the key insights out of that work, and also some additional perspectives on where we are with respect to looking ahead to potential applications for the first gene drive field trials for malaria. So we'll quickly go over a few housekeeping notes. If you encounter any technical issues, please email events@fnih.org. We'll be happy to try to help you out. Webinar audience members, your video and audio will remain off during the webinar, but during the Q&A, we have some ability to bring people in for questions, which we'll manage. And then we would encourage you to use the Q&A function anytime during the webinar to go ahead and add questions that you have. And the speakers will be able to see those and potentially answer them as we go along, but then also we'll have some Q&A time at the end. I'll be happy to give people the opportunity to ask further questions and have some discussion at that point. Okay, so I'd like to introduce today's speakers, and first I would like to apologize for an unfortunate change. We had been hoping that Damaris Matoke from the Kenya Medical Research Institute would be moderating today's webinar. She unfortunately had a conflict come up in her schedule and is unable to join us today. So in addition to opening the webinar, I'll be stepping in as the moderator for today's session. The first presentation that we'll have is from John Connolly, who will begin by presenting key findings and conclusions from the journal article. He's a senior regulatory science officer at Target Malaria. His work encompasses regulatory science, risk assessment, and thinking about how these gene drive approaches could eventually be integrated into malaria control strategies. Then we'll hear from Brian Tarimo from the Transmission Zero project. So he will talk about looking ahead to field trials from the perspective of a researcher, a field researcher who's thinking about that. He is the lead scientist at the Ifakara he is a lead scientist at the Ifakara Health Institute in Tanzania. He specializes in molecular biology and bioinformatics. And the transmission zero approach has been looking at this transmission blocking strategies in transgenic mosquitoes. And he's working on how those could be evaluated in real world settings. And then our third and final speaker, Brenda Das, who's a senior technical expert and policy and regulatory lead at the Gene Community Global Collaborative here at the Foundation for the NIH. She's going to share some thoughts about how this research could be used in this technology, and in particular, what the governance over proposals for initial field trials for these will be. So some of the regulatory systems that are in place and the kinds of approvals that are necessary for this kind of research to move forward in any setting. With that, I think we can go ahead and transition to our first speaker, John Connolly. Thanks so much for joining us today, John.
Pleasure. Are you seeing the correct version, the presentation mode?
Yep, looks perfect. Thank you very much.
Thanks for the introduction, Mike. So I'll begin by just contextualizing why we're interested in field trials. And I wanted to show you very quickly some data that we have from strains that we've been developing for use in gene drive for malaria vector control. And this is a cartoon of a gene known as double sex. And the interesting thing about double sex is it's responsible for sexual differentiation in anopheles. And the gene encodes two transcripts. One is specific for females and one is specific for males. And if we introduce a mutation in the sequences of the gene that are responsible for the female specific transcript, in homozygous females, we see complete sterility, whereas in males and heterozygous females, their fertility is normal. And in addition to that, in the homozygous females, they have multiple anatomical defects, but of importance, the proboscis is disrupted so that they're unable to blood feed. So the homozygous these females with that specific mutation are both sterile and non-biting. And when we went further and didn't just mutate, but introduced a gene drive system at the exact same location, it also caused homozygous females to be sterile and to be non-biting. And when we took that gene drive and introduced it into large cages of wild-type anopheles, what we found was that the gene drive increased in frequency over the course of about a year in a population of around 1,000 mosquitoes until it predominated with homozygous females. And when that happened, there was a resultant drop in the egg output until the populations crashed. So this looks like a very promising approach to what's called population suppression gene drive for malaria vector control, but all of these experiments are in the lab. So the question for us is, what will it take and what is required, and what would we want to evaluate when we think about transitioning this technology to the field? And so the first question is, well, what would the purpose of those field trials be, or any field trials for that matter? And of course, there's efficacy, what the impacts on the target mosquitoes are, but also on malaria transmission. There's also a need to evaluate safety identified from risk assessment in particular. So that would mean impacts on health, both human and animal, impacts on biodiversity, non-target organisms, and the environment in general. And then there's also a need to consider how a trial of this sort might impact on stakeholder perceptions on the technology in general and the trial specifically. What I want to zero in on in my presentation is considerations around efficacy. And one of the defining characteristics of low threshold gene drive is the fact that it has a potential to spread indefinitely and persist indefinitely, which actually makes it an attraction in terms of malaria vector control, because it's an intervention that you can imagine could be quite cost-effective, sustainable, and wouldn't require changes in human behavior, for example. In thinking about gene drive itself, there are a number of categories that can be classified based on whether the gene drive is high threshold or low threshold. And high threshold means a large number of mosquitoes would be required for the gene drive to become self-propagating. Low threshold means that relatively few would be required for release. And they produce either self-limiting gene drive, so that gene drive wouldn't spread and persist indefinitely, or self-sustaining gene drive, where it would spread globally and be persistent. And in fact, the considerations that we're talking about here, we focused on low threshold gene drive, so that self-sustaining gene drive that has the potential to have the most differential impact on malaria vector control in terms of the array of gene drive approaches. But it does come with some challenges around field trial design. And if you're thinking about a first field trial, and shown here in cartoon is a very simple example where we have 2 release sites shown in blue and 2 control sites shown in black, and you get spread of the gene drive over time shown here as clouds. Well, that's fine for the first field trial, but for subsequent trials, given for low threshold gene drive the potential for indefinite spread and persistence, the spillover from the trial could affect subsequent trials. So even with the benefit of potential indefinite spread and persistence, how do we manage this potential for spillover? The WHO is already alive to this issue and has published guidance on use of genetically modified mosquitoes, and including gene drive mosquitoes. And they propose a testing pathway for genetically modified mosquitoes that in phase 2 focuses on entomological endpoints, and then in phase 3 subsequently moves to looking at epidemiological impacts. But they recognize that with this potential for spread and persistence of gene drive, field testing may be better conceived as a continuum of expanding releases. And of course, there are precedents for that. It's not just unique to low threshold gene drive in the context of, say, releases of Wolbachia in ADs or in classical biological control systems. In addition to those considerations, there's a number of points to think about in terms of determining site selection for initial field trials of gene drive that will influence the design. And I won't go into all the detail, but just to say that those factors include operational viability, so how the regulatory structure is structured, as well as whether there's public health surveillance data in existence, but also at those locations what the species composition is, whether there's multiple species of the Anopheles gambiae complex or not, for example, what the statistical power would be, and that would depend on both site characteristics and the ambition of the first trial. Importantly, from WHO guidance perspectives, the first field trials are recommended as having some degree of isolation of the target mosquito population. So that that is part of the consideration for what a trial design would look like. And there's also some more theoretical considerations. There's been examination of potential technologies or even insecticide-based remediation that could be used to make such releases reversible. And related to those and part of the mix is what the strength of evidence might be. And in thinking about, for example, phase 3 trials that look at epidemiological efficacy, there's a number of different trial designs, but a cluster randomized controlled design is considered to be the gold standard in providing the strongest evidence on epidemiological efficacy. However, it's important to remember, even with the potential for indefinite spread and persistence from first releases, that field assessment of novel vector control interventions, I'm thinking particularly of insecticide-based ones, typically start with pilot or phase 2 studies that assess entomological impacts. And so given this potential spread and persistence of low threshold gene drive, there's tensions that need to be balanced between the desirability of conducting those kind of phase 2 trials on entomological efficacy before phase 3 epidemiological cluster randomized control trials, and assessing epidemiological efficacy in cluster randomized control trials before spillover from initial releases might confound data interpretation. And just to contextualize field trials in low threshold gene drive versus insecticide-based approaches, we can think about them and reveal characteristics that are different between both approaches. So insecticide-based interventions, although there might be some effects, there'd be minimal spillover impacts as opposed to gene drive, where we might expect potential indefinite spread. For insecticides, the intervention might persist only temporarily, but could be withdrawn, and that's not the case for low threshold gene drive. Insecticides might act against a broad spectrum of insects, whereas gene drive is designed to target specific populations. However, gene drive would require stringent biosafety assessment, and that's not a consideration for insecticides. Another important difference is the epidemiological impact is more or less immediate from use of insecticide-based interventions, whereas the epidemiological impact from a gene drive approach is expected to be slower over a period of months to years. And for insecticide-based interventions, the phased approach to field trials is well established. And as yet, we've no experience in field trials for gene drive. In terms of insecticide-based interventions, distribution of the intervention is one of the major expenses, whereas for low threshold gene drive, especially at the beginning when we know we have such little data from the field, monitoring is likely to be a major expense. And then finally, something that I want to spend a little bit of time on is the causal pathway, the mechanism of action, if you like, of insecticide-based approaches starts with toxicological impacts on mosquitoes, whereas the mechanism of action for gene drive starts with its own transmission. And so I just want to go through how we expect low threshold gene drive to behave in the field in a little more detail. So what we would first expect is that we'd release the gene drive system, we'd get mating and transmission from the released mosquitoes into wild-type target populations. We'd see the gene drive increase in frequency in those populations at release locations. And allied with that, you'd get a propagation of the genetic traits, which is intended to reduce the vectorial capacity of those target mosquito populations. So for the example that I showed you at first, that would be infertility in our homozygous females. The next step in the causal chain is entomological efficacy, where at a population level, you would start to see a decrease in vectorial capacity of the target mosquito populations on Plasmodium at the release locations relative to control locations and over time. And then finally, the third phase of efficacy in the causal pathway would be epidemiological efficacy, seen as decreased incidence or prevalence of malaria at release locations compared to control locations. And another feature over this causal pathway is the dynamics of how the gene drive will increase in frequency and spread. And in that genetic efficacy phase, we would expect, as the gene drive is spreading through the target population, it's increasing in frequency, whereas once we have impacts on entomological and epidemiological efficacy, we'd expect the gene drive to be at or close to reaching some sort of fixation or equilibrium. So therefore, the causal pathway for gene drive has three principal steps, and testing some or all of these would be a primary objective for the first field trials. And while achieving epidemiological efficacy, the last of the three stages, is obviously the ultimate goal. Even the demonstration of genetic efficacy would be a significant step in development of field testing for gene drive for malaria vector control. So I just want to illustrate what would be meant by genetic efficacy in an actual field trial design. And I'm showing you here a field trial over 4 years, starting off with a baseline. And in terms of genetic efficacies, there's two elements. One is the increase in frequency of the gene drive system at release locations, or the increase in the proportion of mosquitoes that have that gene drive system. And the second feature of genetic efficacy is spread. It's spread from release locations. So in the baseline year where there's no gene drive, we'd have a population of mosquitoes. And here, this is an example that might be more typical of a country like Burkina Faso that we operate in, where there's a highly seasonal trend in the density of mosquito populations. And we'd have a trial design where we had release locations shown in blue and control locations shown here in black. And when we would release, shown here with the gold arrow, what we would expect to see is an increase in frequency, as shown here with the light blue line, over the course of the first year, and that would be simultaneously preceded with some increase in the frequency of gene drive mosquitoes. We'd still see a significant number of wild type mosquitoes, but in the first year, we probably wouldn't expect to start to see impacts on density of the mosquito populations. But at the same time, we'd expect to see the gene drive spread in target populations away from the release locations, shown in panel B below. And then in year 2, this is when we might anticipate starting to see entomological impacts, where the gene drive has reached some sort of equilibrium, at least at the regional level, and then you might start to see impacts on the density of the population of mosquitoes in that year. And in terms of spread, we'd see further spread from the release location, and in a well-designed trial, still not spreading into controlled locations. And then in subsequent years, we'd see continued persistence of the gene drive transgene, much lower abundance of target mosquito populations, and continued spread of the gene drive system. In terms of what entomological efficacy in the same trial would look like, so we'd start off with comparing the abundance of the target mosquito population at a control location, which is shown here in red, versus the population at the release location. In the year of release, we wouldn't necessarily expect to see a dramatic change in a reduction in density of the target population at the control compared to release. but by year 2, we would expect to see that change in density in control locations versus release locations, and that would continue in subsequent years of the trial. And likewise, in terms of epidemiological efficacy, we'd have a similar situation where we'd be looking at in this example, prevalence of malaria at release locations compared to control locations. So release would be purple and control orange here. In the release year, where we see an increase in frequency of the transgene, but no change in the density of mosquito populations, we wouldn't expect to see a change in prevalence. But upon reduction in target population densities, we'd start from the second year expecting to see a decrease in prevalence at release locations compared to control locations that would continue and expand over time. So they're the 3 levels of efficacy that we've dissected that we think will be important to ultimately investigate for any program of field trials for release of a gene drive. And in here, again, I'm always trying to bring this example back to population suppression gene drive. And so, yeah, the ultimate need to evaluate epidemiological efficacy in a cluster randomized control trial is obviously heavily going to influence the objectives and design of what a first pilot trial for low threshold gene drive might look like. And so what I want to end with is just a couple of potential examples that could be explored and that we did explore in the paper. So first of all, given the fact that we have release of a gene drive system that potentially spreads and persists, we could envisage going straight to a cluster randomized control trial. However, although that would provide an opportunity to evaluate the impacts that we're ultimately really interested in, we're still faced with a lack of evidence on genetic efficacy, and in particular, how the gene drive system spreads and the rate of spread. And that might make designing a trial involving clusters, potentially up to 40 or even more, for a typical cluster randomized control trial, very difficult to confidently develop. And that would just give us a high degree of uncertainty over whether the trial could be successful or not. So a second option is to instead have a pilot trial that focuses on genetic efficacy, and that would allow you to obtain information, particularly on spread, in order to parameterize the design, particularly distance between clusters, of a subsequent cluster randomized control trial. However, it could be argued that this could potentially lose opportunities to evaluate epidemiological impacts for reasons of spillover. So a third possibility is to design a scaled, increasingly sophisticated pilot trial that builds into a cluster randomized control trial. So here you'd have a pilot trial that initially focused on genetic efficacy, but that built into it had a number of match pairs where randomized controlled assessment of entomological and epidemiological efficacy could be performed. Then the data on spread would feed into a design of a second stage, much larger trial where there was a sequential addition of additional match pairs. So this obviously has the advantage that it would give us data on spread. It retains the opportunity to look at epidemiological data from the outset, but it potentially has the drawback that you'd have many parallel field logistics, stakeholder and cost considerations that could make the design of the trial quite unwieldy. And then a fourth possibility is to develop a pilot trial in genetic efficacy where, in addition to data on spread, there was some rudimentary data that assessed trends in epidemiological and entomological data that were then used to power statistical calculations for a subsequent randomized control trial. So in conclusion, we've made really significant progress on product development to advance our ambitions for low threshold gene drive. And we think we'll be conducting trials in the next 5 years if we get approval. With this potential for indefinite spread and persistence of low threshold gene drive, that's a strength for malaria vector control, but a challenge in designing a trial. But there are precedents from, for example, Wolbachia classical biological control. Regardless, the primary objectives of the first field trials are going to be to test the causal pathway to efficacy that I described. And field trials will ultimately want to assess epidemiological efficacy in a cluster randomized control trial, but there are options on how we get to that. And I've shown you some of those. And importantly, data from pilot trials where potentially we don't necessarily measure epidemiological data first will still be highly valuable in understanding the rate of spread and variability of spread of gene drive to inform the design of subsequent trials that will measure epidemiological impacts. And so you can do that either in the design, in power calculations, or by directly integrating those data into subsequent RCTs. And I will stop there. Thank you.
Thanks so much, John, for that really excellent survey of many of the important considerations going into the thinking about designing the first gene drive field trials. One of the strengths of this exercise in bringing together 3 different groups that are all looking ahead to potential field trials of gene drive for malaria is the fact that each of these groups is approaching it in different ways. Both the actual mechanisms of the gene drive to reduce the vectorial capacity, but also the potential sites of the field trials and the potential approaches to evidence generation. And so one of the things I think we wanted to highlight through bringing the group together was that there is this spectrum of approaches. This is a field where there's not yet one right answer, as John was just illustrating. A number of different potential approaches could be deployed. And so it's great that we can share some of the different perspectives of how different groups are moving this forward. And with that, I'm delighted to turn it over next to Brian Tarimo with the Transmission Zero group, which is considering many of the same factors that John was just outlining and approaching some things similarly and some things differently than some of the considerations of, say, target malaria. So, Brian, thanks so much for being with us today. If you want to go ahead and come on camera and off mute.
Yes, thank you, everyone. Thank you, Mike, for that introduction, and thank you, John, for that very good presentation. My name is Brian Tarimo. I'm a research scientist at the Fakara Health Institute, and I'll be presenting regarding the perspectives from a project that's operating on gene drive technologies in the field. Next. So I'll walk you through the current landscape of malaria vector control and briefly discuss about why, with the current landscape, scientists in malaria control are looking into gene drive technology as an alternative. And then I'll briefly discuss about phase testing pathways, which John has has highlighted, briefly talk on the regulations and engagement. And I will not do this project any justice if I do not briefly mention about Transmission Zero and how the project is gearing up towards this first initial field trial. Next. So if you, between the year 2000 and 2015, there's been significant reduction in malaria burden and mortality. And if you look at the different interventions that have been used to combat malaria, to control malaria, you can see that vector control interventions such as ITNs and IRS have significantly contributed to this. So in the slide on the left, the red line shows the actual reduction that has happened with regards to malaria incidents, and the line in black shows that they're counterfactual if no intervention were in place between the year 2000 and 2015. And the in-between are the significant contributions between different interventions. And you can see that the contributions from vector control, that is insecticide-treated bed nets, and IRS have significantly contributed to this. Also, in terms of the graph on the right, it shows the cases averted for these interventions. And again, we clearly see that the cases that are averted due to vector control interventions such as bed nets and IRS have significantly contributed to this. Next. However, despite all these successful stories with vector control tools, they're not as perfect as we think they are. If they were perfect, then we wouldn't be talking about malaria, or malaria would not be a significant issue as it is right now. We have facing challenges with insecticide resistance in bed nets and in indoor residual sprays. Screens are imperfect with holes, then they allow insects to pass through. Fumigations, they bring about pollution. Habitat removal, they're challenging. Some are very difficult to find, and when they're easy to find, they're not fixed and findable. So with all these different interventions that we have in the vector control toolbox, we're facing a lot of challenges with them, and therefore this has necessitated that scientists look elsewhere for other interventions that could supplement the current tools. Next. So if we go and look at what makes a malaria such a good vector, factors such as its feeding behavior, vector susceptibility, its longevity, density, meaning the ability for a high reproductive rate, and also its preference for human habitats and host seeking, we can find that almost each and every one of these factors has a genetic component. That means that these phenotypic traits are predetermined by some genetic trait within the mosquitoes. And since that the genome of the mosquito is known, that means we are able to genetically manipulate mosquitoes and therefore kind of create some form of genetic biocontrol for malaria control. Next. And this also is supplemented by the fact that the genome of the mosquito is known. The Anopheles genome has been around for over 20 years, since the early 2000s. And also we have genetic tools such as RNA, which has been around for quite a while, and most recently the CRISPR-Cas9 technology. So all of these knowledge and tools that we have at our disposal enable scientists to be able to genetically manipulate mosquitoes to create for genetic biocontrol. Thanks. Now, if I move on to explain what gene drive technology is, so go next. So gene drive is, as John explained earlier, is a natural genetic phenomena, and it's any genetic element that is able to bias its own inheritance among its offspring. In normal inheritance, which is Mendelian inheritance, there's a 50-50 chance of getting a genetic trait from a parent. There's 50% from a mother and 50% from a mother. But what gene drive does, it biases this inheritance, and therefore there's nearly 100% inheritance of a particular trait from one of the parents. Now, if this technology is coupled to a trait that is able to genetically biocontrol mosquitoes, we're able to select this trait and also to have it increasingly common within a population of mosquitoes. Next. For example, here, if you look from each successive generation, from generation 1 to the last generation, that in each of the successive step or each of the successive generation, we see that engineered or the gene drive, the trait that is coupled to the gene drive increases in frequency. And therefore, this is able to reach fixation. And therefore, when we reach our end generation, we've been able to genetically change the mosquito population. Next. So the different gene drive systems that are being deployed right now, or that are being not deployed, they're being tested. One is population suppression on the left, and this is where you treat the insect with the altered genetic characteristics introduced in the wild population, and through sexual reproduction, through mating, they're able to lead to the suppression of that population. And the other system that's been investigated on the right, the insects with the genetic characteristics are introduced to the wild type, and through mating, they're able to replace that population. And then so you have eventually you have a population of modified mosquitoes. So for population suppression, one of the example is mosquitoes that are sterile. What John was talking about initially in his presentation with the double sex mosquitoes. And an example of the population replacement or population modification is what the Transmission Zero project has been working on, and whereby the antiplasmodium effectors have been introduced into these transgenic mosquitoes, and they block the development of plasmodium within the mosquito, and therefore you have eventually you have mosquitoes that are unable to transmit plasmodium. Next. So phase testing pathways for genetically modified mosquitoes. Go next, please. So also, as John has explained, the causal pathway, once you would want to, in any field trial for this, for the first field trial for this, for the genetically modified mosquitoes or with gene drives, you would want to release gene drive mosquitoes in the target population, and you would want to see that these mosquitoes are being transmitted to subsequent generation. And as you're doing this, you would also want to see that there's an increase in frequency and spread of the gene drive mosquitoes in the target population. And as this is happening, there's also the propagation of the linked genetic trait, which is able to reduce the vectorial capacity against the plasmodium. This would eventually lead to reduced vectorial capacity for plasmodium in the target population, and eventually at the end you will see a decrease incidence in malaria prevalence. So in the first field trial, as these mosquitoes are being released, some of these questions you would want to be answered, and how you go about it, it will depend on the phased pathway that's been which I'll talk about in the next slide. Next. So on the left is the first testing pathway for genetically modified mosquitoes as per WHO guidelines. There's kind of basing on what phase 2 field trials are in pharmaceutical, biopharmaceutical, or pharmaceutical compounds. There are 4 phases. And phase 1, which will be restricted into a laboratory where safety and efficacy will be tested. And then from here, you move on into the field study, which could be either physically confined or ecological confined, for example, in an island. And then you can do open releases. And then there's going to be post-release implementation surveillance. However, because gene drives pose one issue, that they will indiscriminately persist in the population because they are low threshold and they're supposed to spread and increase in frequency. There's not really there's no really clear delineation between phase 2 and the subsequent phases, and therefore there's been some form of modification in the testing pathway, whereby we are able to account for some of the spillovers or these effects that that might happen with regards to this indiscriminate spread and increase in frequency. So that's what the pathway that's highlighted on the right-hand side of my slide. And as the pathway is being this first initial pathway is being conducted, one of the issues that needs to be taken as a priority is the objective. What we want to see from the trial, as also John has explained. Do we want to look at genetic efficacy, entomological efficacy, or epidemiological efficacy? Next. I hope everyone can see this clearly. So based on what is the objective of the trial, we could delineate the phase 2 and phase 2 components, where phase 2A, there's a genetic efficacy study where the genetics the effector of the genetics of the gene-drive mosquitoes are being looked into, and then from there on move to a phase 2B where we assess the entomological efficacy, and then as explained by my colleague earlier, that the ultimate phase of the trial is to look at the epidemiological efficacy to see that there's decreased incidence and prevalence of malaria. Now, this will depend, some of these questions the best way in answering some of these questions will depend on the locations, and the locate will need to be within an island or within an inland, a mainland area. Each of these locations has its own advantages and disadvantages. And within these locations that you choose, which clusters or which areas do you choose to be where you would release your intervention, meaning your gene-drive mosquitoes, or where you would release your interventions? And also some of the considerations that need to be taken into, some of the things that need to be taken into consideration is some of the confounding factors, for example, rainfall or malaria transmission. This might actually augment or obscure some of the comparison between these two localities that you've released your gene-drive mosquitoes. And there are ways to be able to solve or to go around this issue. As John has explained earlier, that cluster randomized control trials has been the go-to for testing a lot of these vector control interventions. But with gene-drive mosquitoes, because of their indefinite spread and persistence in the population, the classical cluster randomized control trials cannot be deployed in this sense, and therefore this will also depend on some of the questions that you want to ask. For example, the genetic efficacy or entomological efficacy or epidemiological efficacy. And also there's ways to adapt to do an adaptive trial design where you could actually introduce some flexibility as the trial process is going along. For example, in the case which I will highlight a bit later for the Transmission Zero project, it's where the effector and the drive have been separated, and this gives you the opportunity to first try the effector and see if the effector does some of these has the genetic efficacy and entomological efficacy, and then perhaps later that could be coupled with a drive, and then that you could be able to will allow you to answer some of the other questions in phase 2B and phase 3B. Next. Next, all of these have to happen with regulations and engagement. There's governance oversights to be able to carry out some of these trials in a safety and secure manner. Globally, we have the Cartagena Protocol, the Nagoya Protocol, and other guidelines and oversight documents that allow for these trials to happen. I've shown some of the documents that we have here in Tanzania, some of the manuals for biosafety, containment, confinement, and for genetically modified organism. And also, next. Next. And also there's stakeholder engagement. Through the stakeholder engagement, this will be able to do to understand the concerns of the community, and also and from these concerns of the community, some of these concerns will be incorporated into the trials to be able to effectively conduct this first trial. And also an effective environmental risk assessment has to be carried out that will be able to address some of the questions that are going to be that need to be answered in this first trial. Next. Lastly, I'll discuss a bit about Transmission Zero. So Transmission Zero is a collaboration of 4 institutions, which is Imperial College London, Ifakara Health Institute in Tanzania, National Institute of Medical Research, and Swiss and Tropical Public Health Institute. Our gene drive system focuses on population modification, as I explained earlier, where we plan to introduce these anti-plasmodium effectors into mosquitoes and for the purpose of blocking transmission of plasmodium. The transgenic work is carried out at Bagamoyo site of Ifakara Health Institute, and the entomological field is expected to be carried out in Ukerewe, which is an island within Lake Victoria. Next. The two antimicrobial peptides introduced are maganin-2 and melittin-2. And initial work carried out was shown that when Plasmodium falciparum blood was spiked with these 2 antimicrobial peptides, there was significant reduction in oocyst intensity within mosquitoes. And some of the preliminary work that has been done with our colleagues at Imperial College London has shown that when these 2 antimicrobial peptides are introduced into mosquitoes and you have transgenic mosquitoes, there's also delayed development of these mosquitoes, and such that the delayed development prolongs the development process of the mosquito, of the oocysts. Therefore, they are now able to be transmitted, to reach the salivary gland and to be transmitted onwards in the malaria cycle. Next. Sorry, next. So briefly to touch on the design of the construct. So the maganin-2 melittin is being engineered into a carboxypeptidase gene within the mosquito. This carboxypeptidase gene has been shown to increase in its expression following a blood meal ingestion. So we've been able to hijack or to the regulatory mechanism of this gene. That means we are doing very minimal modification. We're introducing very minimal modification into the mosquito. There's extra information that shows that we are only 800 kb has been added into the mosquito to create this transgenic line. Next. So I think I'm nearing the end now. So this is the phased approach that the Transmission Zero is hoping to take. So the following laboratory studies to study the efficacy and safety of the mosquitoes, and also moving to semi-field and large cage studies and semi-field studies to show that some of the efficacy and safety concerns with these mosquitoes. We plan to release only the effector strain in small isolated release. This will be able to allow us to look into the genetic efficacy to compare how do these mosquitoes fare compared to wild-type mosquitoes. So looking at genetic efficacy and some of the entomological efficacy. And thereafter, we plan to release also in small-scale open release is to look at to release these mosquitoes with the effector molecules and also with self-limiting source of Cas9. This will also ensure localization of these mosquitoes, and also we'll be able to test some of this genetic efficacy, entomological, and some of the biological efficacy. And later on, based on this data, is to move on to large-scale open releases, where we'll release both the mosquitoes with the effector trait, and also with the gene drive, with the Cas9 trait that's capable to drive. And this will be able to inform us on all the 3 genetic, entomological, and epidemiological efficacy. Next. With that, I hand it back to you, Mike. Thank you very much, and thank you for your attention.
Excellent. Thank you so much for that, Brian. Very much appreciate that perspective, and again, highlighting, especially at the end there, some of the diversity and potential approaches that the different researchers are taking to field evaluation of the constructs in different locations and in different kinds of genetic approaches as well. So for our final presentation before we get to the Q&A session, I'm delighted to hand it over to my colleague, Brenda Das. And already there's been a little bit of discussion in the Q&A, and I would encourage people to, as Brenda is speaking, add questions for her, add questions for the previous speakers. We can answer some of those in the chat, and then some of them in the Q&A after Brenda is complete. So Brenda will present a little bit more about some of the governance questions over the potential for future field trials for gene drive. Thanks, Brenda. Okay, slides look good.
Okay, so like Mike said, thank you for joining us today, everybody. And like Mike said, I'm going to talk about regulatory considerations. Let me jump ahead. So I just want to start by saying that engineered gene-drive mosquitoes are being developed as a vector control strategy to be used within existing integrated vector management. So whatever the trials are designed for these technologies, they should be designed to reflect this use case within IVM protocols that are in existence and in use. So vector control is at the heart of a One Health approach to disease interventions and can impact in all areas, including public health, veterinary health, and ecosystem health. But today's topic is the most proximal use of gene drive technologies, which is for malaria control and elimination. And I just want to share that these technologies, as they come to field trials, will be undergoing multiple levels of oversight from multiple different entities at various levels. And I'll talk about some of these entities in some detail today. So Brian has already set the stage for you, but in case you didn't realize this, the reason we're talking about countries in Africa is that Africa has the highest burden of malaria. And as I mentioned, the most proximal use case for the technology is for malaria control by engineering mosquitoes. So moving ahead, I want to jump right ahead into regulation, and I'll start with a top-down approach, so with the highest levels of reach of the jurisdiction. So I'll start with international considerations, and here I'll talk about the UN conventions. This is of importance because we're talking about malaria in Africa, and most of the 55 nations that are part of the African Union have signed on to various UN conventions that regulate work with GMOs. Primary on this is Cartagena Protocol, which is a protocol which supplements the Convention on Biological Diversity, which is a UN convention. Cartagena Protocol itself deals with living modified organisms that are results of modern biotechnology. Living modified organisms are essentially genetically modified organisms. And to clarify, engineered gene drive organisms, which would include engineered gene drive mosquitoes, are considered LMOs and therefore would be under the scope of this protocol. This protocol also looks primarily at the safe transfer, handling, and use of LMOs, especially if they are able to have an adverse effect either on the conservation of biological diversity or the sustainable use of biodiversity. The Cartagena Protocol itself has an annex within it, Annex 3, which covers the basic principles of risk assessment, and countries that sign on to the Cartagena Protocol, otherwise called parties, are obligated to follow through on all the parts of the protocol, which includes this risk assessment. And essentially what they do is they bring this into their national legislation and regulations so that they can be in keeping with the terms of the protocol that they've signed on to. There are two other supplements to the Cartagena Protocol. So there's the Nagoya Protocol on access to genetic resources and the sharing of benefits accrued from these resources, and the Nagoya Kuala Lumpur Protocol on liability and redress. These may have an impact on the research and development of engineered gene drive mosquitoes. So moving to another level of international consideration, this is the World Health Organization. Brian and John have described some of this work, and I'm showing you here 3 documents that are reports or guidances from the World Health Organization that cover engineered gene-drive mosquitoes and also genetically modified mosquitoes. I will clarify that GM mosquitoes is an umbrella, and engineered gene drives would be a type of GM mosquito. So it would extend to engineered gene drive mosquitoes. And what you can see here is the phased pathway that is described in the guidance framework for testing gene modified mosquitoes. And it shows that phased pathway exposes the environment and humans to the mosquitoes as you go from contained to field release. And so as containment decreases, obviously exposure increases, and regulatory oversight starts right from the lab and goes through to field release and post-market. So in terms of these engineered gene-drive mosquitoes, because they are a new technology to the world and to WHO as well, they would probably, in all likelihood, come to the new intervention pathway. And you see this pathway here on the right. It essentially is a pathway that has already tested pesticides and other technologies, insecticide-treated bed nets. There are some GM products that have been considered by the Vector Control Advisory Group, or VCAG, such as the sterile insect technology that Oxitec uses, as well as some work done by IAEA. Essentially, the WHO looks to assess the public health value and then come up with a policy recommendation, which involves 3 different advisory groups. And then it's a soup to nuts approach. So they also are interested in looking at product quality, safety, and efficacy, and will also inspect the manufacturing facilities if there are any manufacturing facilities. And once they come up with both those work streams, they will, if the data is convincing and supportive, qualify the product and list it. And this is important, again, for African countries, because if they want to purchase these products, it helps if it's listed by the WHO for the nations to procure them. Scaling back down from international to regional considerations, I'm sharing here the EAC medical medicines regulatory harmonization scheme. And the reason why I'm focusing on EAC is because it is a regional economic community of 8 countries that was pilot for this medicines regulation harmonization framework. The goal was to take a regional approach for clinical trials and medicines, new medicines being brought to Africa, and then scaling from the East African region to other regions, as you can see across the continent. And since EAC was a pilot, in the last 10 years, they have shown a robust methodology and framework to have common technical documents that are based on international documents such as the WHO pre-qualification mechanism and the ICH to register medicines. They have now a good common information management system to bring the dossiers through, the applications through. They have also established quality management systems, inspections, and are working on also a pharmacovigilance strategy. So the point of this is to say that there are structures in place, and this regional framework has now included 5 different regional economic communities and will soon hopefully spread across the continent. So there is the ability to take on medical product clinical trials. And of course, these clinical trials will not be one-to-one with engineered gene-drive mosquitoes, but they will probably use some of the same frameworks and some of the same institutional systems. So I'll describe some of the potential for this in the next few slides, but keep this in mind. And looking at another one that's not that dissimilar is the West Africa IVM platform, or the West Africa Integrated Vector Management Platform, that has been set up by the African Union Development Agency's NEPAD program. And this is, again, a regional program specifically built to bring on board new technologies for integrated vector management, such as the engineered gene drive mosquito. And it's focused on West African states. So it looks at the economic community of West African states, which, or ECOWAS, which is also a regional economic community, and uses the West African Health Organization as a secretariat. And what you see on the slide are documents that they have put together as guidelines for a variety of topics that would impact the development and testing of engineered gene drive mosquitoes. So they have biosafety guidelines, institutional biosafety committee training, risk assessment of LMOs, and the other documents that are also required to streamline the initial processes of contained research and initial genetic testing. Coming down one more level to the national level, this is just a graphic to show you the different types of organizations that a developer would encounter. So when an applicant is interested in doing any research with an engineered gene drive organism in Africa, they typically, this is the structure that they would follow. They would bring their application to an institution where they want to work or partner with, and it would go to the Institutional Biosafety Committee, which is associated with that research institution. The primary objective of the IBC is to ensure that the research is conducted safely. And so they have to ensure safety to the researchers, the people that work in the lab, and the people in the community surrounding that institution. And then once the IBC has evaluated the application, the dossier that comes with it, which would also have an environmental risk assessment, they will send this application to the competent authority for that country. So this is a federal enterprise, often called a National Biosafety Authority or a National Biosafety Committee, depending on the country. And since this is a federal organization, it is under the oversight of a ministry. And the ministry may differ depending on the country, but it could be the ministry that undertakes science and technology, or agriculture, or environment. And it may also have other ministries that share oversight depending on jurisdiction and the laws in the country. But then that ministry is the one, like I mentioned earlier, taking the CBD priorities, because if the country has signed on and is a party to Cartagena Protocol, then those the intent of those international treaties must be reflected in the law of the country. So the ministry in charge would be the one that would implement that law. So this is the structure in most African countries. Obviously, there might be some subtle changes, differences depending on the country, but in most countries, this is how it would go. Now, for actual field release and testing, this slide is meant to indicate that what John and Brian have told you is basically aligned with the testing guidance that was described in the WHO framework. And as you see, going from left to right, you're going from most contained to least contained. And essentially, this is no different than the drug paradigm that is used globally. So you go through initial design discovery, preclinical, to lab animal or some sort of animal model, and then eventually to healthy patients, and then to the patients that have the condition. And if you have enough data that supports your claim, then an entity in the country or an international entity, both with the entity in the country, will allow for registration of your product, and then you release your product into the market. So I'll go into each phase that John and Brian described. So phase one would be contained. It would be your research in the lab. Essentially, as I mentioned, in country, the developer, the applicant, would submit a dossier, including a risk assessment, to the Institutional Biosafety Committee, who would then survey it to make sure that it was everything that was needed by law, and then would submit it to the National Biosafety Authority, or the competent authority in the country. And then, as I mentioned, there would be a parent ministry that has policy oversight over the NBA. There might be other ministries that would also have an input. And then you would also have other bodies that would inform the NBA, including inspection and monitoring authorities. And then there would be some form of public comment at both the IBC level and the NBA level for this work to be done. And the decision making would happen at the NBA. The decision would be communicated back through the IBC to the developer. This decision would have the research permit in the terms of the study, the terms that are enclosed in the permit for the scope of the work. In terms of what the developer would have to provide, it would be a dossier that describes the work. It would be a risk assessment, as I mentioned, and also probably a detection method for the engineered gene drive mosquito. In case there was any escape, they would use this method to validate that it was a mosquito that had the engineered gene drive or not. There may be a request from the IBC to have the facility inspected. There may be a requirement for the facility to be certified. And if any of the engineered gene drive organism material needs to be imported, there would be the need for an import permit. So moving on to phase 2A, if you're doing early genetic studies, so if you're doing small cages or large cages, you would still have essentially what I showed in phase 1. As you move towards release, you would look to design a study where you were testing the efficacy of your engineered gene drive mosquito. Most of the primary outcomes or endpoints for your study would be entomological. So does the transgene spread? Does the transgene remain stable in the population, in the mosquito? You would need to, again, go through the pathway that I mentioned. For phase 1, you would engage the IBC and the NBA and multiple ministries. Potentially, if you are doing, open release or large field cage studies, you may need to also have a consultation with the Environment Ministry or the Health Ministry in the country. You would now be providing a monitoring plan, not just for the transgene in the mosquito, so the transgenic mosquito, but you would also be looking at certain aspects of the environment, monitoring them as required by what the findings were of your environmental risk assessment. In some countries, you may also need to do environmental risk assessment and an ESHA, which is socioeconomic and health impact assessment. You would, as usual, with any work with LMOs, have to provide a remediation plan, stopping plan in case there were any adverse events. And you would be doing stakeholder engagement at both the public, local, national, and regional levels in the event that the trial was designed so that it spread, the likelihood of spread to other nations or other jurisdictions was high. So you would be looking at transboundary effects, and hopefully the regional level regulation would help with navigating this aspect of it. So moving from phase 2 to phase 3, you would then also be going to do epidemiological endpoints. So the goal of your clinical trial or your field trial would be to include human subjects. You would be looking at the disease incident in the area of your trial. So you would probably need to also engage with ethics committees, the research, different institutional research boards, and also your national ethics committee and data safety monitoring board. So now you would definitely be talking to your health ministry in country, potentially also regionally, as I mentioned, with the medicines regulatory harmonization. If you're doing a field trial for human outputs, you would be talking to the clinical trial boards in those countries. Potentially also, as it applies to insects, I put GCP and GMP, but these are not medical products, so you would probably have to have some form of facility inspection. It would not necessarily be GCP or GMP, but it would be something in the spirit of the same. You would have to design your clinical trial, have it approved by the countries, and register the clinical trial. So there are clinical trial registration platforms in Africa as well as in other countries, depending on what the country requires you to do. And for your monitoring plan, you would now also be monitoring for the public health endpoints that the country requires you to monitor. Your remediation plans would now include human health and public health endpoints, and you would have to also consider standard of care. So as I mentioned, engineered gene drive mosquitoes are being used along with the existing IBM, in the same spirit, you would also be doing a trial with the existing standard of care for the malaria patients. Your trial terms would be such that because engineered gene drive mosquitoes are designed to spread and disperse, that they are likely to start small but eventually hope to scale up. So the trial would be designed in such a way, and the regulation should follow suit, where there would be hopefully interim checkpoints, and that if you reach the endpoints for those interim checkpoints, you would be allowed as the applicant to scale up your trial. That would be ideal. And that may not happen, but that would be ideal. Again, transboundary effects as you scale up are expected. And so the approval coming regionally would definitely be of use. Data sharing regionally would definitely be a big plus. You may also have to consult with the WHO, and it would be good to, from the WHO's perspective, if you did also think about doing a cost analysis. And this is just to show you what I've already said, that there is a drug approval process that is a robust system in East Africa. Again, I'm focusing on East Africa because this was the pilot and they have the most experience because they've been doing medical clinical trials for about a decade now and have a well-established framework. They have clear timelines. It's a very transparent system. And I show you Kenya, but the way that the MRH works in East Africa is that you submit to Tanzania, and then Tanzania shepherds it through the process, and different aspects are taken on by different countries in East Africa. I'm just showing you this because it spells out more the ethics committee and how it's built into the system of the clinical trial protocol evaluation. And as I mentioned, there is a good timeline. It's very transparent, and all the forms and the procedures are available online. So one would hope that they would be in a good shape to take on an engineered gene drive mosquito as well, and look at the human disease endpoints for this when the time comes. Phase 4 is in the event that the trial meets its goals and all stakeholders are satisfied with the results, then you would hope that it would go to post-market performance and monitoring, where your mosquito just continues to spread and disperse, and you continue monitoring for safety reasons and surveilling for things that you need to surveil for in the environment as well as with public health. So you would still be monitoring for the transgene in the mosquito population. You would still be monitoring for environmental endpoints that came out of your ESHA as well as your ERA, public health, and any socioeconomic considerations that came out of your ESHA findings. You would be doing surveillance for adverse events, and you would have an idea because you've been doing this monitoring for a while at this point. So it's not like you wouldn't have data already. And then you would check back with the regulators to see if you need to change your monitoring plan, either change what you're monitoring for or the frequency, or even drop some of the aspects if you don't see any adverse events. The rest of it remains the same. You would be doing, the stakeholder engagement, public communication. And at this point, transboundary effects are a known entity because you're doing it at a regional level. The only thing I'll say is that if you're not the only engineered gene drive mosquito in the area, then there would be some amount of work to do on product interaction if there's more than one engineered gene drive mosquito in the field at that point. But this is quite far down the road. And by then you would have a lot of experience, a lot of data, and a good idea of how the mosquito spreads and what sorts of downstream effects happen from its spread, both for the environment and for the disease itself. So I end here just to show you that it's not happening in a vacuum, that the work is well regulated, and there are structures in place on continent, in the countries, transparent frameworks, timelines, and expertise. Thank you.
Thanks so much, Brenda, for that review of the different layers and different kinds of governance mechanisms and regulatory reviews that would be in place before any of the kinds of proposed field trials that we've been talking about here would have permission to move forward. We have 6 or 7 minutes left before the end. Really appreciate all the questions that have come in the chat and the activity of our panelists in addressing those. Would welcome any additional questions that anyone wants to drop in. While we're waiting for that, I thought I would pick up on one of the threads from the Q&A, which is, you know, at the outset, you know, John, you outlined, and the paper outlines, that there are a number of different considerations for how to move forward with the first gene drive field trials. And there are lots of different kinds of pros and cons for different approaches. There's, you know, there's not just a single clear, this is obviously the way that everybody should do this. And so Brian and John, could you both talk a little bit about how within your projects you're approaching, you know, how to navigate the different options that are available to you? You know, one of the specific questions from Jason Delborn was, you know, to what degree does external consultation about the different options that you have inform the way that you're moving forward? So maybe, Brian, if you want to start, and then John.
Yes. Yes, thanks, Mike, for that question. So what we've done as a project is, one, we've started a lot of engagement with communities in the areas that we plan to do these releases. There's a stakeholder engagement team has planned several activities with the communities so that they kind of understand what the project is about and what the eventual trial will be about. And this, we've not only stopped there or done that, we also, there's a lot of engagement with regional and district leaders, so local government, in terms of the local government within the plant field area. There's also been a lot of discussion and communication with the regulatory authority. So in Tanzania, the biosafety is under the mandate of the Ministry of Environment. So there's been a lot of discussion with that ministry. All the approvals, all the dossier and documentation applications has to go through them. So there's been that discussion in order to kind of see once we reach that stage, what kind of trial we plan to do. I hope if I've missed anything, maybe John can add into it.
Thanks, Brian.
That's a great answer, Brian, that we've similarly been engaged with a large stakeholder engagement teams in Burkina Faso and Uganda in particular, where we're active in GM contained use work or field releases. And probably one thing that didn't come across enough in my presentation, in the spirit of trying to squeeze everything into 20 minutes, is that we're not the decision makers here. So we're the proposers of options and potential ways. And we've really been talking about, or I've really been talking about ways to measure efficacy in the field, but it won't simply be an internal conversation in Target Malaria about what our field trial will be and what, from the design to the implementation, there's going to have to be a lot of dialogue and engagement and readjustment to reflect the views of, on the official level, regulators that will make decisions about approvals to conduct field trials, but a wide array of stakeholders that clearly have important contributions to make in what the scope of a first field trial should look like. And included in that, I would also say funders, because these trials are expensive to do. Even the less ambitious and more straightforward ones are expensive. So trying to find a way that addresses any concerns that people have, but also is robust enough to produce data that will give meaningful information on whether this intervention can be tested further or is efficacious is going to be absolutely crucial. And that, yeah, all I can say is that will not be something that will happen within any one project. It's going to have to involve a lot of discussion in the broadest of communities.
Thanks so much, Sean. And then maybe for the very last quick answer, some of the questions, I think essentially related to questions of risk management. So Brenda, could you just give a minute on how, of course we talk a lot about risk assessment, prospectively trying to identify risks, but about risk management planning in this process?
Yeah, thanks, Mike. In terms of risk management, once the risk assessment is completed and submitted to the National Biosafety Authority, I'm talking about the early stages, so the contained research, the IBC and the NBA will look at that risk assessment and make a determination of whether there is residual risk that they are concerned about and that they want mitigated. And if they want it mitigated, then they will have to work with the developer to come up with mitigation strategies. So whether that includes monitoring peripherally around, let's say, around the facility, capturing mosquitoes standardly to make sure that, what's caught doesn't have it, and then testing it using a validated method to show that it doesn't contain the transgene, to determine whether the containment level is multiply sufficient. And then there's also all the physical restrictions of containment. So these are all risk mitigation strategies, at least in the containment side of things. Once you do field release, risk mitigation looks different because it's no longer about containment, because the whole point of releasing is to get it out of containment. So there would be other ways of testing, of bringing back some level of safety in case of adverse events. So you would have to predefine what an adverse event would be in this case and what it is that you would need to do to mitigate that adverse event. So you do a pathway and determine if this event could happen, how likely is it to happen? And if it's likely to happen, then let's say you apply more insecticides, in case of that event happening to get rid of the mosquitoes. At this point, there's not an easy way to address, dampening the gene-driven mosquito, maybe release, sterile mosquitoes to outcompete. There are other ways to do it. So those would be risk mitigation methodologies in the field if you got to a field release. There's also geographic isolation and other features you could use. Do it in, a low density season, so there's less mosquitoes out there. Obviously, all of these would affect your output and your ability to make a determination of an effect size that had value. So it's a checks and balance kind of a question and would depend on so many variables, including what the appetite for risk was at the national and local levels.
Excellent. Thanks so much, Brenda. Okay, well, again, thank you to John, Brian, and Brenda very much, and thank you very much to everyone who's attended today. We really appreciate this opportunity to share this work and these perspectives with everyone, and all of the questions and interactions in the Q&A. This is obviously an important ongoing topic in this field. There will be many more opportunities to engage and discuss. We really appreciate it, and we welcome anyone reaching out about this at any time. So again, thank you very much. Take care. Bye-bye.
Bye.
Bye.
Bye.